Hello everyone, I will present our work towards a multi-omics workflow. So to start, actually in a cell we have several very important biological actors such as DNA, RNA or proteins, and in fact now with bioinformatics we can have thousands of values for them. So for instance with genomics, transcriptomics or proteomics. So our work was focused on the development of new methods to identify and handle multi-omics signatures towards a better biological understanding, and to develop a software with a graphical interface to be used by biologists. So to define some important terms, by multi-omics I mean an experimental context where for the same samples we have thousands of features for different biological actors, and where we have for the same patients or samples labels. More by molecular signature I mean a minimum set of features enables a good classification and understanding. So for our work we can imagine RNA-seq or RPP data. So then we carry out feature selections with differential expression analysis with DSEQ2 or LEMA for instance. So we have a minimal list of features, significant ones, and with them we can carry out functional enrichment with overrepresented analysis or GSCA for example. So we get one enrichment table results for each omics block, for RNA-seq, for genes expressions or protein expressions. To explain what's an overrepresentation analysis, in fact it consists in detecting pathways potentially changed or modified between two phenotypes. In fact, we carry out an overrepresentation analysis on only one block of biological data. For instance, here the proteins one. Then we perform an differential expression analysis to have only a subset of proteins, selecting significant one. For example, we can get 50 proteins. In the other hand, we have pathway information. We can retrieve this from wiki pathways, gene ontology, reactome databases, and so forth. A pathway database consists in a file with the pathway IDs and the genes that are part of them. So to carry out another representation analysis, we harness this data, computing a proportion test called also an exact feature test. In fact, we want to answer the following question: which are the pathways with a significant number of differently expressed genes? Let's represent this method with an example. So we can take our 50 differently expressed proteins. Then the proportion tested are proportion of differently expressed proteins in the pathway, 24 here, and differently expressed proteins not in the pathway, 26 here, out of all pathway genes number and all proteins from biological data. So with the Fisher test we get a probability value, and for this given pathway, so for all pathways in the database we get a probability value, and so the result of another representation analysis, so of the functional arrangement of these proteins, are a table with all pathway IDs and their probabilities. So for all omics data block, so proteins, genes for instance, we get one table. So we get several tables in a multi-omics context. So in our work, we merge these tables with a method, with a probability poolings method. In fact, to boil down over represent analysis results, we use Stauffer's method. So we have RNA, proteins, or microRNA, for instance, and we carry out overrepresent analysis to merge enrichment results. So what is Stauffer's method? In fact, you have 3 tables of enrichment results. So with ID for pathways and their probabilities. You want to merge it, so you use a standard normal cumulative distribution functions to, in fact, retrieve z-scores from probabilities for each probability you have. So for 3 probabilities, we get 3 z-scores, new z-scores, and we can also, before to merge it, add weights, so such as proportions of features involved in pathway enrichment, and with the sum of the z-score weighted, we can get only one z-score from 3 z-scores, and so we can retrieve only one probabilities from 3 probabilities. So after this, we carry out an enrichment map that enables visualize our results, our enrichment table results from Stauffer's probabilities pooling. In fact, an enrichment map can be built with enrichment map cytoscape module. This is a network where nodes are pathways, so pathway you have in the enrichment results table. More one edge between 2 nodes is drawn if genes are shared, and nodes are then clustered according to network topology in biological themes. So with this workflow and used on breast cancer data from lobular and ductal phenotypes, with genes and proteins expressions from TCGA, and used with and enriched with pathways for reactome, we have this network. So we can see this network is really highly dense. It's hard to interpret. Actually, we have a lot of nodes. In fact, we have more than 200 significant pathways, and there are not pathways very specific to non-differences between lobular and ductal phenotypes. So it's hard to interpret with this kind of workflow. So we propose to replace the differential expression analysis by multi-omics 1, named SPLSDA. So we use SPLSDA from Diablo framework from Mixomics Error package. In fact, it's a multi-omic regression with a sparse dimension to select features and a discriminant analysis ones. This is based upon dimension reduction with linear recombination of features. And these methods enable feature selections more stringent, with 50 genes, for instance, for SPLSDA, instead of hundreds or thousands for differential expression analysis. And these features are with a high classification power, and these features have classification power, and they are more correlated between blocks and two phenotypes than differential expression analysis. So we assess if this change makes biological interpretation easier. To have a quick sight of what SPLSGA does, I display an example with two blocks of data, the green and the red ones, with two features, A and B and C and D. When we talk about linear recombination, we can think about principal component analysis, so PCA. And here I represent linear recombination as the green stick and the red stick. In fact, in our case, we can compute correlation between news values of this linear recombination. So with R as coefficient of correlation of 0 here. And what SPLSGA does, in fact, is as the animation shows, is adjusting linear recombination according to correlation between news values after recombinations. More, we can carry out feature selections based on the values of the coefficients of the linear recombinations. In fact, so we will use the same workflow after. So with functional enrichment, with overrepresentation analysis or GSEA, with Stauffer's probabilities pooling, and with enrichment map building. So with this workflow, we get this. We can see this workflow enables a network not highly dense. And in fact, we can see biological themes such as overexpressed ERBB2 or organizations of cell junctions. And in fact, we have only 50 pathways, and with biological themes, biological clusters, very specific to differences known between lobular and ductal. So using SPLSGA instead of differential expression analysis seems to make interpretation easier with few clusters, a few amount of clusters, and highly related to lobular and ductal phenotypes. For this work, we embed this analysis in a R package accepted recently in Bioconductor project. It's named multi-site, and we have a graphical interface to make SPLSGA selection method, overrepresent analysis, Stauffer's method, and to reproduce this workflow. So to build on our work, we illustrated multi-omics interest to study complex phenotypes, such as breast, lobular, and ductal phenotypes. We've assessed probabilities pooling by Stauffer's method and the use of enrichment map. More, we have shown that feature selection based upon differential expression analysis gives enrichment map highly dense and hard to interpret, contrary to feature selection based upon SPLSGA that shows great improvement for biological insight discovery with pathways correlated to differences known between both phenotypes. More, we have also assessed feature selection based upon multivariate methods such as SVM or random forest that gives highly dense networks such as differential expression analysis. It could be interesting to assess others multi-omics or multi-blocks feature selection methods or feature selection models that enable feature selections, such as RGCCA, for instance, to see if this is specific to SPLSGA or if we can have this kind of results with others multi-omics analysis. So thank you for your attention, and I will be happy to answer your questions.